Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm very excited to be here today with Mike Snyder, who a great majority of you all will have either read his papers or come across him a variety of times. He's the director of the Center for Genomics and Personalized Medicine at the Stanford School of Medicine, and he has done some seminal work in this space over the past decade. So, Mike, it's really excited to be here with you today.
My pleasure.
You are famous for monitoring health in an incredibly detailed and multi-omic way, and perhaps also very famous for doing so on yourself as participant number one in many cases. I'd love to just start there of how you got interested in this topic and what are some of the big things you've learned over your career, either about yourself or about multi-omics and precision medicine more broadly.
Sure. Well, I guess it depends how far back you want to go. But if you go back early enough, I'm dating myself, but I came out of an era where it was one gene, one PhD, the recombinant DNA era, where people would clone a gene. And if they got a lot of characterization done, that even made their PhD a little better. And then I guess our shtick was that this seemed a little, strange. And why don't we try and get all the relevant genes or proteins at once? So we came up with a scheme. I was working on yeast at the time of tagging all the genes and studying these particular cell biological problems, cell polarity, chromosome segregation. And again, not the one gene at a time, but get all the genes and characterize them. And then about 15 years ago, I moved to Stanford. And so we were studying that. By the way, that sort of launched the field of systems biology. And then we always wanted to apply it to medicine. I was on a main campus at Yale, which is a great place, but it was hard to do medicine when you're on a main campus. And then I moved to Stanford 15 years ago, embedded in the medical school, and the idea was we could then apply the same concept, if you will, to medical problems. And really, that's how it worked. Just start getting big data around medical problems. And I guess if at the risk of rambling on a bit, it seemed like, why don't we start with this precision health idea? Could we, I think it always bothered me that we were doing sick care rather than health care, and should we really be collecting deep data around people, just like we're doing around biological systems? And so when I moved to Stanford, actually, believe it or not, but when you go to a new place, they take blood from you, right? You get in a new health plan and they load you up. And I said, well, can you give me a few extra tubes of blood? And this is a story I don't tell very often, but that's really how it launched. I knew we wanted to do this. I said, well, while I'm here, can you give me a few extra tubes of blood? And we process it. And that really put them in the freezer, and then, got that going, basically, this whole concept of deep data around people. And I've always viewed it best to start with me, when we do these things, to see what I can learn and this sort of thing, rather than some average person who, you might screw it up and waste their samples, this kind of thing. Whereas I can always go back to a doctor and bleed myself or something.
That's amazing. And you've looked over the years, you've looked at microbiome, smartwatches, continuous blood glucose monitoring, obviously genetics. What have you found to be the most impactful or informative, and maybe in combination as well? Probably a big theme of your work is that in isolation, none of these tools are necessarily enough, but in combination, you get some really interesting insights.
Yeah, that's exactly right. I might even give you a little history. It started with the omics because we were very versed in that. Our lab invented a number of these technologies, RNA-seq, things like this. And so we're very comfortable. We're a little unusual in that. Also dating myself a little bit, people were expert in nucleic acids or proteins, and we were, because of the way we rolled things out, we kind of got expert at both proteomics and nucleic acids, so, RNA-seq, this sort of thing. And then metabolomics, we weren't, but we picked that up pretty quickly when I moved to Stanford. So we were collecting, we'd start with the omics because that's where I was most versed. And then actually out came the wearables as like fitness trackers. And we said, well, gosh, those are pretty cool health monitors. Let's start throwing them on our cohort. We were setting this group of 109 people. And yeah, and in the end, I mean, to your point, I would say the wearables are a very, very high impact for low cost. I mean, you may know right away we discovered you could tell when people are getting ill. It's sort of with my Lyme disease. We picked that up asymptomatically from a smartwatch and a pulse ox. And so we realized that you could start telling these shifts from baseline. That's a big theme of our work. Get deep data dives and know what your baseline is so that you can see these shifts. And that's how I figured out my Lyme disease. And then an obvious follow-up was we saw these glucose monitors coming out being used for type 1 and insulin-dependent type 2 diabetics and said, well, let's start putting these on normal people and see what they look like. I think that, again, that's our big theme. What does a so-called normal healthy person really look like? And then you discover right away, well, normal's not so normal, that there's plenty of so-called healthy people that are spiking just as bad as type 2 diabetics, have no idea about it. And it has to do, we think diabetes is extremely heterogeneous. So you can pick up these things that would get missed if you're just looking at hemoglobin A1C or fasting glucose, this sort of thing. So the bottom line is that, again, and that's always been a big theme of our work, let's not make assumptions about things that we think we know already. Just start diving in and see what things really are and let the data tell us what's going on rather than come in with these preconceived hypotheses.
What is the story about detecting your Lyme disease for people who haven't heard it? That's an amazing one. I have a Whoop, and I am a big proponent of keeping track of things and seeing when my pulse starts to go up or respiratory rate increases, probably getting sick. But Lyme disease is a whole different story. What's the background?
Well, backing up a little bit. So we're keen on wearables. You can see my 3 smartwatches. I normally have 4. My Garmin broke, so right now I'm down to 3, and I have an Oura ring. Believe it or not, I normally wear hearing aids, which are sensors too, and they're actually pretty useful because you wear them all the time. I wear them for hearing, of course, but they're also good monitors as well. So anyway, those are the different devices, and the story behind the Lyme disease was that I was actually, the backstory was helping my brother put up fences in rural Massachusetts for one day. And as you may know, in Massachusetts, 55% of ticks were Lyme infested at that time, maybe higher now. I don't know. And then, 2 weeks later, I was flying to Norway, and I measure myself all the time. That time, they didn't have pulse oxes on smartwatches, so I was putting that on my finger, and we had discovered that your blood oxygen drops on airline flights. That's known already, not well known. Most pilots know it. Most flight attendants don't, it turns out. Most people don't. That's why you get tired on airplanes. So it's just, I'm measuring myself all the time. I saw my blood oxygen, but this time it dropped really low. We know how low it goes for particular kinds of aircrafts and altitudes. And so it went unusually low and it never came back. And I also noticed my heart rate jumped up as well, and that also never came back. And I didn't notice at the time, but my skin temperature was higher too. I just didn't catch that. And this is all pre-symptomatically. And then I did get symptoms off and on, but I could tell something was off again, and I had the data to back it up.
Yeah.
So I did go to a physician in Norway. I warned him it might be Lyme because of that 2-week window in there. And he draws blood and sees my monocytes, my immune cells are up. Says,
wow,
you've got a bacterial infection. And here's another lesson. He wanted to give me penicillin, and I said, no, I need doxycycline, which is what you take for Lyme. And, it was a little tense. I didn't speak Norwegian. Turns out he spoke fine English. So I brought my wife. She's my witness. She was there. And he, in the end, gave me doxycycline and cleared it right up. And when I got back, I got tested. I was Lyme positive by an antibody test and even had some antigens. And because I measure myself a lot, I'd given blood 3 days before I'd left. And sure enough, I was negative. So I seroconverted during that time.
Wow.
So the point out of the story is that pre-symptomatically, I could see when I first got Lyme just because of this jump up in heart rate. And then we went back. I had 2 years of data. So I looked at, all the number of times I was off, and I was off 4 times. And I give blood a lot and measure myself a lot. So One was a Lyme time. There were two viral infections when I saw this jump up in resting heart rate. And then there was a fourth time I was asymptomatic, but I had high C-reactive protein, which is a sign that you're ill. And every single time I was ill by CRP, these other things, I could see this jump up in heart rate. So we wrote an algorithm to tell when you're getting ill because of this jump up in heart rate. And it worked on me, again, pre-symptomatic, but because we had this group of people, it worked on every one of them, always in advance of symptoms. It's a very sensitive measure.
Yeah.
Yeah, so we published that in 2017 and, quite proud of that paper. And then once you know it, out comes the pandemic in early 2020. And we had been improving the algorithms, this sort of thing, and we were pretty well prepared. We partnered quickly with Fitbit. And, first step was can we tell when people are getting COVID in advance of symptoms? And sure enough, the algorithms we wrote, we could see that you can, same thing works, that your heart rate jumps up in advance of symptoms. And for COVID, it has a long pre-symptomatic period. And so it's actually a really great detection system, works 80% of the time. We've written a real-time detection system, works 80% of the time, not 100% because we don't get a stable baseline on everybody. But we're trying to improve that. I know with more data, we can get that higher. But anyway, it did work. We could tell, and we had this alarming system. We loved any listeners want to sign up. It's innovations.stanford.edu/wearables. Do feel free to sign up for our study. We want to perfect this because I should say it's a general stressor, meaning it turns out the number one trigger of red alerts is workplace stress.
Sure.
Mental health.
Can imagine that.
Yeah. Yeah. And other things will trigger it too, but most of those are contextual. Like if you run a marathon, your heart rate will be up for several days, and therefore that'll trigger red alerts. But you know what's going on there. That's not an issue. But so I would say if you're just sitting around and you get one of these red alerts, you're either mentally or physically stressed. And we think that's useful. Either way, we want to know that. And with further refinement of our algorithms, I know we'll tell the difference between mental health stress and a respiratory viral infection.
You mentioned this interesting idea that there are people who are diagnosed as being diabetic, but then there are many of us who have, maybe similar level of insulin spikes or other kind of indicators, but aren't diagnosed. What do we know about what proportion of the population is in those different states? And also, what is the long-term impact of health? Because I've got to imagine that if you're in a kind of cryptically diabetic state for a long period of time, that must correlate with some kind of negative health outcomes. So I'm curious on what you've learned about that.
Great question. Well, a couple of comments there. First of all, people who start getting these spikes, we think will go on to be diabetic. And a good example is gestational diabetes, right? Women who get gestational diabetes during pregnancy with a high probability will become diabetic later in life. So it's kind of like a precursor signal. That's how we view it. No data to prove that yet, although it is the case that people who are pre-diabetic will become diabetic. And you can also get a correlation between the amount of glucose spiking, if you will. It's called time in range, and relationship to hemoglobin A1C, a measure for diabetes. So there are these correlations. But even more than that, to directly answer your question, there are correlations between these postprandial spikes, I call them these post-meal spikes, glucose spikes, and cardiovascular disease and several other serious conditions. So you really do want to get this under control. I think it's missed in part because diabetes is a very heterogeneous disease. People don't realize this. Sure, the major form is insulin resistance, but there's other forms as well, meaning you don't, even for type 2, you may not make insulin, or you may not release it from the pancreas. So just for the listeners, I happen to be a type 2 diabetic. I'm a weird one. If you looked at me, I'm not overweight at all. But it turns out, I'm an interesting scenario, by the way. My genome predicted, actually, Atul Butte, UCSF, now at UCSF, Stanford at the time, he predicted my diabetes. And then after a nasty respiratory syncytial virus infection, I became diabetic, which I discovered because of all this monitoring. So I caught it right away. I got it under control, at least initially. Did come back. And then now I'm on drugs to keep it under control. But my diabetes is a very interesting form. I don't release insulin from the pancreas. So I make insulin, my cells respond, but I don't release it from the pancreas, which is not common, but not unheard of either. It does happen. and I respond to certain drugs that they're called Prandtl derivatives, rapinolide. I respond well to. It turns out I'm not the most common drug, metformin, I don't respond to.
Right.
We like to think of this as precision diabetes, knowing exactly what's wrong with you
Yeah.
to get the right drug. Now, you may argue I'm on an Ozempic kind of drug. It's called Trulicity from Lilly. These are these GLP-1 things you
Yeah.
may have heard of that cause weight loss, which I'm trying not to do, by the way, because I don't
Right.
want to lose weight. I'm fairly thin already, but they work incredibly well. I went from 8.4 hemoglobin A1C to 6.3, just like that. It's incredible. And you may argue maybe we don't need these precision drugs, but I think we still do, because for some people, and I'm a good case, I get a little bit of nausea from that, and I'd prefer not to have the nausea.
Yeah, sure.
And do things by combinations, by control. That would be better. So anyway, I'm probably rambling a bit, but you get the idea. We think diabetes is heterogeneous. And so I think it's missed in some cases because your form of diabetes doesn't match as well with standard tests like fasting glucose and things. But you can pick it up when you do more detailed measurements of the sort we do.
Interesting. When a tool Butte made that prediction, is it monogenic? Is it polygenic? How does the genetics underpinning this actually play out?
Yeah, great question. It's apologenic. So he had at the time the best database for this stuff. And so he was making polygenic risk scores. And I'm one of the few cases where that works. So you may know the polygenic risk scores, they work for the extreme end of the distribution, the top 2 to 4%. Well, that's me. I'm actually in that. So it happened to work. They don't work for most people. We think that's because of the way polygenic risk scores are calculated. For one thing, their linear addition of these variants for the people who follow this sort of thing, that is, and genetics isn't linear. Anyone who does genetics knows 1 + 1 is not 2 in genetics often, right? You have 2 mutations, say biochemical pathway, 1 + 1 is 1. You have 2 mutations of different pathways, 1 + 1 can be 15 in effect size, very synergistic. So we think this idea of doing linear addition is it's fine for a first pass, but it's not going to cut it when you're trying to get predictive models. And it also, these polygenic risk scores, they ignore the rare variants, which probably contribute as well. So I don't think we have the perfect method for analyzing genomes. I do think we now have AI-based methods, which are not linear, that have turned out to be really, really good for gene discovery, and in some cases for setting up predictive scores. We're not there yet, but we're getting better at this on the prediction side. And so I think we should be able to get better at predicting from complex disease, which we're not right now. That's I think an important area that needs to be brought under control.
Yeah. I like you, I'm very excited about the future where it's healthcare prevention, prediction rather than sick care. One of the there are a couple, I think, issues that people run into. One of them is cost of implementing these kind of multiomic programs. And the other is, I guess the precision recall challenges. How much are we spending on the people who are actually fine to find the ones who are really not fine, and we can pick it up? How do you think about what the big barriers are from getting some of the work you do into mass healthcare, where we really can think about shifting the whole system in some way?
Yeah, great question. So what we do as a research project, don't get me wrong, RNA-seq, microbiome, all these things, and these cost millions of dollars, right? This is ridiculously expensive, but it's research. And what we're trying to do is figure out what parts are most important and then how to scale it. Now, I'm a believer academics, we're good at discovery, we're good at proof of principle, we're no good at scaling. And you can take the very best academic labs who think they're good at scaling, and I would argue they're not very good when I compare them to my companies. So we spin off companies, and here I'm 100% conflicted because we've spun off quite a few over the years, but I think that's how you scale. And so from the big data standpoint, we set up a company called QBio. I don't know if I'm allowed to say all these things. But
Yeah, of course you can.
Okay. Anyway, this company, they do a medical version of what I just told you. They don't do RNA-seq because no physician knows how to do this, but they do whole body MRI. Now, any physician today will tell you, don't do a whole body MRI because you're going to pick up these incidentals like these nodules and things. And you ask me, I say 100%, yeah, you should do whole body MRI. The issue isn't, do you have any nodules? The issue is, do you have any growing nodules? And you learn that from longitudinal measurement. You'll never, and that concept seems to be, not in medicine today. Like they rarely look back at your past measurements when you
Mm.
get, go in for a test. So we figure out what's most important, and then we try to actually roll that out. And so they've been very effective, just like the research study. The first 109 people we studied, there were 49 major health discoveries in the first 3.5 years, like early lymphoma, we call it precancers, 2 serious heart issues, etc. And then from this company, same thing, they caught ovarian cancer, prostate cancer, all early, and even early pancreatic cancer, which is almost never discovered early. So these deep data dives, they give you a much, much clearer picture of your health, and you find these things before symptoms. And you might think they'll freak people out. They don't. That's the thing. They're giving you a good exam to see what's going on. All right, something looks off. Let's do some follow-ups. And that's true for our research. It's true for QBio. And these tend to be life-saving things. And I think that's what we got to shift to. So I'm a believer scaling it. And the wearables is a good example. That one we just put out there. Now, lots of companies have these stress indicators on their watches.
Yeah.
It's not at the level I want to see it, but at least it shows the proof of principle. We spun off a company that does CGM monitoring. It's called January Eye. They're trying to get this glucose control measurements out to people. Now CGMs are over the counter, so anybody can do this stuff. And then lastly, we've been doing a lot with microsampling. This idea, little pricks of blood, mail it in, measure, in our case, thousands of metabolites and proteins. That's in the lab, but in the company, they'll measure 600 metabolites called IOLO, and they'll make these deep measurements, and that's not very expensive. So again, that's how we're trying to roll it out to the bigger world. Now, the cost issue, QBio is not cheap. It's $3,500 right now, although I think it can get cheaper in the future, and the other ones are much cheaper. I think, they always start out expensive and the wealthy then pay, but once you show utility, I hope we can get this into the system. Now, there is still one more problem. I know I'm rambling on a bit again, but which is nobody pays to keep you healthy. That's the problem with our current framework in medicine today. And that's why we do sick care, not health care. So we've got to change, the whole incentive structure so that they get aligned so that we can keep people healthy rather than always just treat them when they're ill. And I think there's only two things that motivate people. One is money and the other is family. And so if we can set financial incentives up, hey, how about if you get a discount on your health plan or your insurance if you wear a smartwatch? I think you should get paid to get your genome sequenced because you will take better care of yourself with that information. So we need to restructure this, and that's not easy to do while these things are expensive. So A, we got to get them cheap, which I think we do. And then B, we've got to get people just more aligned.
Yeah, that makes total sense. It seems like the approach with QBio, for example, you can imagine it's $3,500, like you said today, but the more of these you do, the more the cost comes down, the more data you get that can help you figure out what you can strip out cost-wise and still get really good outcomes. And you can see almost the way that Teslas were $120,000 or more originally, and then over time they've just gotten more and more available. I can see this going a similar direction.
Exactly right. Yeah, that is the goal. And then the other is to push this stuff into the home so people do get themselves measured more frequently. The wearables are perfect because you can measure yourself continuously. And I think if we could get the culture set up that people wear these for, again, their health and not be discriminated against, I think that'll be very powerful. I want to take it further, like you say, these little drops of blood that you mail in and get yourself measured more frequently. This idea of going to a physician every 2 years or something, even that's not done usually very often. When you're young, you never go to a doctor. You think you're invincible, you'll live forever.
Yeah.
But I think as you get older, you go, but not that often. And I think depending what your trajectories look like, right? If you start seeing your lipids or your glucose going off, well, go get yourself measured. If it's a home test, it's trivial to do that stuff. And so, especially if these things become over the counter. So I think we can bring health into a more active role while people are healthy, and this health measurement part. And then we've got to get this concept of trajectories and longitudinal profiling incorporated into healthcare as well.
You published some really interesting work recently that I wanted to talk about, which was around aging. We've had a couple of people recently talk about aging. It's a big topic. What I gathered from the paper was that many of us think about aging as a kind of gradual process, and in some ways it is. But some of your recent work actually found that it was punctuated at certain points in our lives, mid-40s and around 60. People undergo rapid changes in their biological aging, but that these are also kind of qualitatively different in some ways. I'd love if you could talk a little bit about that. And were you surprised by that? And how did you find it?
Yeah, sure. Well, I guess maybe a little backstory here is that because we're tracking people over time longitudinally, we can see how they're changing. And we discovered, a few years ago that everybody's aging differently. So some people, we call them cardioagers. Their heart pathways are changing more, cardiovascular disease pathway. Other people are metabolic, other people are immune. We call these ageotypes. And we think that's really important to know exactly how you're aging because there's actionable information associated with this that you can change. So that was the first phase of the story. And I still think that's an important concept, the idea of personal aging. And it is, and the fact that it's different between people. And then the more recent one, we said, well, let's look at the group as a whole, and are there some times when people where there's more changes occurring than others? And as you point out, we discovered two sort of bursts of changes, one in the mid 40s and the other in the 60s. Now, the one in the 60s totally expected because that's when people become at high risk for disease.
Yeah.
We know their immune system declines. That's why people get vaccinated, because one of the things we saw was your viral infection pathways, changing at that time. So
right.
It's a no-brainer, and we know you need to get vaccinated for flu and now COVID as you hit your 60s for these obvious reasons. So that one we saw. We did discover some new things, like we saw kidney function change and carbs change. I think carbs change throughout, but there's a special burst, I would say, as people hit their 60s. And so, again, some of that is useful. Drink more water as you hit your 60s.
Yeah.
We saw muscle mass and skin changes. Well, visibly, you can see that as well. Muscle mass, well known. But the one in the 40s, to me, it was surprising. I think in hindsight, you can see evidence of that out there. People have a tendency to pull muscles. It's true for me in my 40s. We also saw skin changes. And again, this group will understand, but we saw changes in your extracellular matrix and some key muscle proteins showing up in the blood. And so those are probably the reasons why some of this is occurring. We're seeing some of these key proteins. But then we saw other things changes. Lipids were shifting. And I think that fits with the idea that people's cholesterol can start going off. We saw about, your LDL shifting. And I think fat deposition has a way of changing as people hit their 40s. So in hindsight, some of this makes sense. And then we saw alcohol and caffeine metabolism shifting as well, which, again, for me in hindsight, that made a lot of sense because I certainly became more sensitive to caffeine as I hit my 40s. So anecdotally, I think we'd seen this, and I get a ton of emails. Oh, I saw this too. So, but I guess it's nice scientifically to see that, these biochemical shifts are occurring. And I think they're nice because you can, again, act on it at a personal level, like the alcohol. Maybe you have to back off on a drink as you start hitting your 40s or something. And for me, I stopped drinking coffee after noon. Now I even try and stop a little bit earlier in the day because I have become more sensitive. So I think, and the lipid one we think is particularly important. I was kind of slow in actually going on statins myself. And I kind of, in hindsight, I wish I'd started earlier because I actually have a high calcium score because of this. So that's calcified arteries there.
Right.
So that's something that, had I started on statins earlier, maybe I wouldn't have that. Anyway, but the point is now we can become aware of these things. We and physicians can become aware. So maybe people jump on it right away and don't let things, go off the rails. And that's the key to health, as act early, you can solve problems. Act late, you got serious issues. Cancer being the prototype case. If you wait till cancer metastasizes, it's, doomsday. It's not good.
Yeah.
You can act early. You can, in many cases, manage, if not cure. So we think that, that's the whole key to health monitoring, catch stuff early.
On the cancer topic, what's your perspective on cell-free DNA and some of the work that's happening there over the past few years. Obviously, it's the same dichotomy that we discussed before. The detractors say that we're picking up a lot of people that are, otherwise healthy and getting scared or costing the system money. But on the other side, people are also being picked up with cancers earlier. I wonder what your work or perspective is on that.
Yeah, I view all these things as screening, even our own work, right? You
Yeah.
get a first indication. Don't panic, see what's there. And so these tests, I've done Grail myself twice. There, I have no conflict. And, I came out clear, I'm happy to say. But now they are better for late stage 3, stage 4. And it depends on the cancer. They have some power at the earlier stages, depends, as I say, on the cancer. So I still think it's valuable. I've never seen a situation where more data has hurt people, meaning you can see what's going on, then do the right follow-up test. Now, I suppose if you're an anxious person who would be concerned, you want people waking up every morning saying, am I going to get breast cancer? Am I going to get breast cancer? That's not a healthy situation. If you're one of those, you probably want to be, I think you still want to get measured, but somebody else gets the info and then runs the right test for you when you're in for your visits, something like that. But I think most people can handle this information. And the analogy I like to use a lot is, our health is like a car. Your car, it has a lot of sensors on it, and you relay information back to your dashboard. And so that's what we want for our health monitoring. We want a dashboard for our health so we can see when things shift off. You don't panic, but much like your car dashboard, you need to know when you're running low on gas or speeding or whatever, or the check engine light goes on. And that's how I view health. See if a check engine light goes on, do the follow-up test. And if there's something there, you got to deal with it early rather than wait late. So
we can measure so many things right now, from obviously, you're in your research and your company's full body MRIs and imaging, genetics, RNA, proteomics, metabolomics, watches. Are there things where we don't have good measurements yet? Are there big gaps where you'd say, I wish we could measure here, but it's, we're in complete darkness or near darkness around this particular topic? What's missing from the technological toolkit, if anything?
Mental health. It's unbelievable how
we
we do not have good markers for mental health, depression, anxiety. These are generally done by surveys and things, which is a terrible way to tell if somebody's going off. And imagine you're treating someone. Well, how do you know if they're getting better? You run surveys. And that's just not the way to do this stuff. And so I just came out of a neurodegenerative disease meeting, an Alzheimer's and dementia meeting. And same problem there. They, even though they have a little bit now without the amyloid for Alzheimer's, they really don't have it for most things. So we really need good markers for mental health. And so we're actually doing a lot now trying to get markers, digital and biochemical markers. We'll see how well we do. But we should be able to do this. And so I would argue that's an area that we should be handling much, much better than we do. And other areas on the physical side, I think we can still get better on our early detection on cancer. I'm excited about the cell-free stuff, even though people do worry about false positives. But we do need to get better at the earlier stuff, and I think more markers may help. I'm not an imaging guy by nature, but I do think these imaging technologies can be done much, much better than they currently are. And again, I think the one thing that maybe is missing the most is putting it all together. So one thing we discovered both from our research and QBio is that no one measurement really tells you what's going on. That's a theme that's come out of our work. When you see 3 things shifting, let's say even all of them shift a little bit, well, something's off. No one marker is usually that strong an indicator, but when you get the more complete picture, you can really see what's going on. And so I would argue that this integrative notion, and if you think about healthcare today, maybe to rail on a little bit more, is when they give you your data back, you get these sheets of stuff about ions and all this stuff. It's not organized in any useful fashion, right? For either the physician or for the patient.
Yeah.
So how do you, they don't put it around themes. So you have to scramble through, your calcium's up on this sheet, and then you go over to the next sheet and these other cells are up. And it's like nobody put it all together to say, well, gosh, here's what's going on.
You're doing good. Yeah. Or you're not. Yeah.
Yeah, exactly. This is what, and this is, to be honest, pretty simple, but this is what QBio did. They put this in a very nice fashion. They just put it around themes that anybody, a physician or a patient, can understand. And then you see all these things at once too. You look across 5 markers and you say, all right, oh yeah, that something's off there.
Yeah. How much do you think that our own willingness to look inward is the problem here? You mentioned that people don't see the doctor often until it's too late. And I wonder if there's a big factor here of we kind of, sometimes we're blissfully ignorant of what's going on in our own bodies, right? Until things start to go wrong. Is that an element that's at play here, potentially?
It is. I think, and that's human nature, right? That people don't want to know bad news, right? You don't want to know you're at risk for breast cancer or something. On the other hand, you got to know, right?
Yeah.
And so I think we've got to get a cultural shift here so that people are comfortable with it. Assume you have something going on. You are going to die of something.
Yeah.
So you should be tracking yourself. And that is a cultural shift. Just like your car, you don't drive your car around for a dashboard because you want to be ignorant. Well, you want to, drive around in your health and know as much as what's going on so that you can actually see what's happening there. And then we've had people come out of, you can tell I do sell the stuff to my friends and things because I am a believer. And I've had people come out of QBio and said, I'm so relieved I did this because now I see I'm fine. Or even if they have, oh yeah, I saw this thing. I was kind of expecting that. Now I'm going to do the following follow-ups. So you can actually get some assurance as to by just knowing what's going on. I think that's the right mentality we need. Not that, oh, I'd rather not know, but rather I'd like to know. And if something's off, here's what I'm going to do about it. Because I think all problems are solvable. I'm probably one of the few people who thinks we can and live forever, actually.
Yeah.
If not with we're going to need some re-engineering of our bodies to be able to do that, by the way. But I do think that is theoretically possible.
Yeah, interesting. What does your family think of all the monitoring? Do they get involved too? Do they check in on it? What goes on there?
Well, early on, they rolled their eyes, and it's that's their strange dad kind of thing. And it's kind of interesting because my youngest daughter now has a Fitbit.
Yeah,
and it took. Yeah, I don't know how much they want this all disclosed, but anyway, they're starting to ease their way into this.
that's great. Yeah, it is. It's funny. We gravitate towards or away from our parents at different times in life, right? Sometimes it's not, and nothing is forever. As we wrap up here, are there any big themes or areas that you're looking towards for the future for your group or companies, or what you're focused on more widely that you think would be interesting for people to know about?
Well, as you can tell, I'm just so keen on this health monitoring. I really want everybody on the planet to do it. They don't have to be as extreme as me, of course, but to be wearing smartwatches to catch when things shift off early is a no-brainer in my mind. And so I'd like to see us culturally adopt this idea about tracking people while you're healthy.
Yeah.
I really think we're going to live longer, healthier lives. And maybe to this point, I'm sure you know this, and the audience knows this already, but the difference between people's health span and their lifespan is 11 to 15 years, depends how you measure it. And so that means they're living their last decade of life unhealthy. And that's just not a good situation to be in. It's why we spend so much money on healthcare. U.S., as everybody knows, spends way more than anyone else. And we're doing a lot of that in that last year of life, these last few years, when people are extremely unhealthy. And so I honestly think we can keep people healthy all the way to the end. And that is the mission. Just keep everything going and working right. And I think, my own experience is just talking with people are happy to live forever as long as they're healthy. And it's only once their health starts declining, they're kind of getting into this mindset, all right, well, maybe my time has come. And then, they hang in there for another 8 or so years. And I just think we can change this whole culture and I guess that's my mission. I know this is probably pretty Pollyannish, but I really think we can keep people healthy mentally and physically for a long, long time by just monitoring and taking action.
Yeah, it's an amazing North Star. Before we started recording, I mentioned to you how 11 years ago or so, I was actually in your office before I went and did my PhD and was just asking advice on what kinds of things we're going to be interesting in the future. I've always found you incredibly inspiring. That was an inspiring conversation. This one was as well. So I just want to say thank you for the advice 11 years ago. And also thanks for joining us again today.
My pleasure.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible. In particular, Amy Cousins and Sonia Shah, who produce the show, and James Pierce from Selective Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.